Good morning everyone. Today I, Saumya Bhardwaj, Research Scholar of Geology Department of Dr. Hari Singh Gaur Vishwavidyalaya, is going to present the video on the multiple sequence alignment. Multiple sequence alignment and its various applications in bioinformatics. Multiple sequence alignment are an essential and widely used computational procedure for the biological sequence analysis in molecular biology, computational biology, and bioinformatics. Multiple sequence alignment is a collection of three or more proteins or the nucleic acid sequences that are partially or completely aligned. Homologous residues are aligned in column across the length of sequences. Aligned means arrangement. Residues are homologous in an evolutionary sense. Not only in evolutionary sense, but residues are also homologous in the structural sense. Multiple sequence alignment are completed where the homologous sequences are compared in order to perform a phylogenetic reconstruction, proteins secondary and tertiary structure analysis, and protein function prediction analysis. Biologically, good and accurate alignments can have a significant meaning, showing various relationships and homology between the different sequences, and can provide useful information which can be used to further identify new members of protein families. The accuracy of multiple sequence alignment is of critical importance due to the fact that many bioinformatic techniques and procedures are dependent on multiple sequence alignment results. Due to the multiple sequence alignment significance, many multiple sequence alignment algorithms have been developed. Unfortunately, constructing an accurate multiple sequence alignment is a computationally intense and biologically complex task, such as no current multiple sequence alignment tool is likely to generate a biologically perfect result. Therefore, this area of research is very active, aiming to develop a method which can align thousands of sequences that are too lengthy and produce high quality algorithms, and that too in a reasonable time. Algorithm speed and computational complexity are negatively affected when the number of sequences to be aligned increases. The recent advances in high throughput sequencing technologies means that this sequence output is growing at an exponential rate. The biology landscape being punctuated by a number of large-scale projects such as a Human Genome Project, 1000 Genome Project, and a Genome 10,000 Project. Indeed, technologies such as Roche or 454, Illumina, and Solid are capable of producing the gigabase pairs per machine per day. The entire worldwide second generation sequencing capacity surpasses 13 base pairs per year and is continually to increase yearly by a factor of 5. Other large-scale data is emerging from a high throughput technologies such as a gene expression, datasets, protein 3D structures, protein-protein interaction, and others, which are also generating huge sequence datasets. The analysis and storage of growing genomic data represents the central challenges in the computational biology today. As the protein alignment problem has been studied for several decades, studies have shown a considerable progress in improving the accuracy, quality, and the speed of multiple sequence alignment tools, with manually refined alignments continuing to provide superior performance to the automated algorithms. However, more than 3 sequences of biologically relevant length can be difficult and time consuming to align manually. Therefore, computational algorithms are used a matter of course. Sequences can be aligned in two ways, either globally or locally. Sequences can be aligned using their entire length, that is the global alignment, or at a specific regions, that is the local alignment. Multiple sequence alignment for protein sequences is much more difficult than the DNA sequence equivalent containing only 4 nucleotides due to the fact that there are 20 different amino acids. Global optimization techniques developed in applied mathematics and operations research provide a generic toolbox for solving a complex optimization problems. Global optimization is now used on a daily basis, and its application to multiple sequence alignment problem has become a routine. Local alignments are preferable, however, they can be challenging to calculate due to the difficulty associated with the identification of sequence regions of similarity. The two major aspects of importance of multiple sequence alignment tools for user are the biological accuracy and computational techniques. The biological accuracy concerns how close the multiple alignments are to the true alignment and are the sequences aligning correctly, showing various insertions, deletions, or gap in the right position. Computational complexity refers to the time, memory, and CPU requirements. Complexity is of increasing relevance as a result of increasing number of sequences needed to be aligned. Multiple sequence alignment is almost the most useful tool in bioinformatics as it helps in almost every application of bioinformatics, predicting the protein structure, predicting protein function, phylogenetic analysis, etc. Now, after discussing about the multiple sequence alignment, I will discuss about the applications of MSA. First, structure prediction. A multiple sequence alignment can give us the almost perfect protein or RNA secondary structure. Sometimes it can also help even with the 3D structures. Second, protein family. A multiple sequence alignment can help us to describe our own protein is a member of known family or not. Third, pattern identification. By looking at the conserved regions or sites, we can identify which region is responsible for a functional site. Fourth, domain identification. By looking at the file provided by a multiple sequence alignment, we can extract profiles to use them against the databases. Fifth, DNA regulatory elements. We can use multiple sequence alignment to locate the DNA regulatory elements such as the binding sites, etc. Sixth, the phylogenetic analysis. By carefully picking up related sequences, we can construct a tree using the sequences that we have used in the multiple sequence alignment. Seventh, multiple sequence alignment is more sensitive than the pairwise alignment to detect the homologs. Eighth, population data can be analyzed in a multiple sequence alignment, that is by the pop set. Ninth, blast output can take the form of multiple sequence alignment and can reduce the conserved residues of motifs. 10th, a single query can be searched against a database of multiple sequence alignment. The best example is PFAM. Now, last but not the least, regulatory regions of genes may have a consensus identifiable by the multiple sequence alignment. I hope this video was informative and you all have liked it. Thank you very much.